All right, so we'll start with the definitions. So starting with dominant, how would we define something that is dominant? Right, perfect. Good job, Tim and Jared. Yeah, so this is going to describe a trait that masks the expression of another trait if both versions are present. Okay, and we'll talk about that in a second. So a dominant trait is always going to mask the expression of something else. Perfect. Good. Then we have recessive. So how would we define something that is recessive? So yeah, so a trait that can be masked, right? So in order for us to see a recessive trait or a recessive phenotype, we need two copies of the recessive allele. Okay, it's not enough to just have one because the dominant one will mask it. Okay? So you need two copies of the recessive trait or the recessive allele in order to see its effects. Perfect. Good. All right. Next one. So phenotype. Okay. How would we define a phenotype? All right, good job, Hasmin. So this is the observable traits expressed by an organism. Good. Perfect, Hasmin and Kimberly. Good, very good. So the observable traits, that's going to be the physical appearance of that particular trait. Good. Okay, what about a genotype? Okay. What is a genotype? Right, the underlying genetic makeup, right? So the genetic makeup is going to be the genotype. The genotype is going to refer to the two versions of the alleles that are present, and that will give the genotype there. Okay. Now, when we're looking at the phenotype and the genotype, does the phenotype determine the genotype, or does the genotype determine the phenotype? Which one determines which? Right, Kimberly, good. So the genotype will determine the phenotype. Very good. Yeah, so genotype is going to determine your phenotype, which makes sense because your genes, your DNA is going to determine what your physical appearance is going to look like. All right, good. Okay, so now going to homozygous. How would we define something as homozygous? Right, good job, Mario. So having the same alleles, right? Having two copies of the same allele for a given gene, right? So if you have big A, or little a, those would both be classified as homozygous genotypes. Okay. What about heterozygous? Right, having two different alleles. Perfect. So having two different alleles, where is that at? Here it is. So having two different alleles for a given gene, right? So if you have a dominant allele and a recessive allele present in one single individual, okay, that individual is considered heterozygous. So their genotype might be something like big A, little a. Okay. Perfect. Any questions on any of these definitions so far? From dominant, recessive, phenotype, genotype, homozygous, heterozygous. Any questions on any of those? Okay, cool. All right, so next one is a test cross. So what is a test cross? Right, it's going to be a cross between a dominant expressing individual of unknown genotype and a homozygous recessive individual, right? So the goal of a test cross is to of a dominant expressing organism. Okay, so remember, when we're looking at something that is expressing the dominant trait, it can either be homozygous dominant or it can be heterozygous. Okay, but we have no way just by looking at that organism Okay? We have no way of knowing whether it's heterozygous or homozygous dominant. Okay? So in order to figure out what that is, we're going to mate it with an organism that we know is homozygous recessive, okay? And then look at the combination of offspring that result. Perfect. Okay? And so the offspring will help us determine whether that dominant expressing parent was homozygous dominant or heterozygous. All right, good. Okay, so that's that. How about codominance? What is codominance? What do we think? Anybody? So not one of two variants of a gene. Anyone have an idea? So codominance. Yes, perfect testing. So it's where we have simultaneous expression of a gene. Where did I see that? Right. So, oh, just kidding, not that. Here we go. So in a heterozygote, we have two copies of the gene, or two copies of an allele that both get expressed, right? So in codominance, for example, you have two alleles, okay, two different alleles, but both of them show up in the offspring. Okay, so for example, when we were looking at last week, the chickens with black and white feathers, right? If you have a black feather allele and a white feather allele, okay, and both are present, okay, you might have a chicken that's got both black and white feathers. Okay? Or if we were looking at codominance, when we're looking at blood types, okay, for people who have type AB blood, you have one copy of the A allele and one copy of the B allele, so they end up with the A and B proteins on their red blood cells. So in codominance, you have a heterozygote that has both alleles being expressed at the same time. Perfect. Okay. Does that one make sense for people? Any questions on that one? Cool. Okay. Okay. So the next one is an allele. Okay, so what is an allele? So it's not the observable traits in an organism. Yeah, so this is the one where you have one of two variants of a gene. Perfect. So one of two variants of a particular gene. So for example, an allele might be, a purple allele, or an allele for purple flowers and an allele for white flowers. Two different variants for the same gene, which was the flower color. So an allele is just a version of a particular gene. All right, and then the last one is X-linked. Okay, so what does it mean for something to be X-linked? What do we think here? Yes, so it's perfect. Okay, good. So everyone's getting this one. So it's going to be a gene that is present on the X chromosome, but not on the Y chromosome, right? So we call that an X-linked chromosome or an X-linked gene because it's only located on the X chromosome. Perfect. Okay, any questions on any of these definitions before we go forward here? Okay. All right, so we'll go ahead and keep going here. So question 2 says, when Mendel looked at the colors of the flowers in his pea plants, what was the ratio of purple flowers to white flowers in the F2 generation? Was it 3-to-1? Was it 1-to-2-to-1, where you have one purple flower, two light purple flowers, and one white purple flower, or white flower? C, 100% purple flowers, or D, 100% white flowers? Right. So in the F2 generation, he saw a 3-to-1 ratio, okay, where he took some heterozygotes of purple plants, or heterozygous purple plants, made them with one another or self-fertilized them. And then he saw a 3-to-1 ratio where he started to see the white flowers start to reappear in the second generation. Good. Okay, good. So number 3, so here we've got a bunch of Punnett square questions. So number 3 says, in humans, the gene for curly hair, HC, is incompletely dominant to the gene for straight hair, which is HS. People who are heterozygous, HCHS, have wavy hair. So Thor and Jane Foster both have wavy hair. They decide to have a baby. What is the probability that the child will have straight hair? Okay. So we'll go ahead and kind of work all these out, and we'll do the Punnett squares for these. Okay, so everyone's already getting this one. So this is 25%. Right, so we'll go ahead and just run through this Punnett square real quick. Okay. So both of them are heterozygous. Okay. We're going to see HC over here, HS on this side, and then again, HC, HS. Okay. So here we would find HC in this top left box. Okay. In the top right, we would find HCHS. Oh, this is bad. Okay, then we have HCHS again. And then the last box, we have HSHS. Right, perfect. So based off of our Punnett square here, hey, we want to know how many, what's the probability that our child will have straight hair? And we only see one of our 4 boxes having the HSHS genotype. Okay, so as a result, we'll only have a 25% chance of having an offspring with straight hair. Okay. So that's what we'll end up seeing here. Cool. All right. So Stephanie, Jared, and Excel, very good on getting this one. All right, cool. So we'll go ahead and do the next one here. Okay. So this next one is in camellia flowers, petal color is determined by codominance. Okay. So when a red flower is crossed with a white flower, FW, all of the offspring are covered in both red and white petals. Okay. So you decide to cross a red flower with a red and white flower. So which phenotypes could be present in the offspring? Okay. So again, we'll go ahead and do the Punnett square to help us answer this question. I'll do this in a different color on this side. Okay. So if we have a red flower, okay, then its genotype is going to be FR. Okay, again, this is determined by codominance. Okay. And then if we have a red and white flower, its genotype is going to be FRFW. Okay. FRFW. Okay. So again, if we do our Punnett square, here we have FRFR. Okay. Top right box, again, FR. Oh, geez, FR. Bottom left, we have FR, FW, and then down here we have FR, FW again. Okay. All right, so based off of our Punnett square, okay, what phenotypes could be present? Okay. Option A says flowers with all red petals and flowers with red and white petals. B, flowers with all white petals and flowers with red and white petals. C, only flowers with all red petals, or D, only flowers with all red and white petals. Okay. So based off of our Punnett square, what should our answer be over here? Yes. So Yetzi says A, flowers with red and red petals and flowers with red and white petals, and that is absolutely correct. Right. We're going to see 2 of our 4 boxes having completely red petals here, and then 2 out of our 4 boxes having a combination of red and white petals. Perfect. So that would be our answer there. Okay. Question 5. Let me clear all this. So question 5 says, in horses, the allele for straight hair is dominant. The allele for curly hair is recessive. So Billy has a female horse that is homozygous recessive for the hair gene. Okay, so it has curly hair. He mates this female with a horse that has straight hair, but he doesn't know if the male horse is homozygous dominant or heterozygous. All the offspring from this mating have straight hair. So perform a test cross to determine whether the parent was homozygous dominant or heterozygous. Okay. Is the male parent homozygous dominant or heterozygous? Okay. So again, we'll go ahead and do our Punnett square here to help us answer this question. Okay. We'll do this in blue. So Stephanie says that our offspring, or that the parent is going to be homozygous dominant, and that should be what we see here. So let's go ahead and do this and work this out real quick. Okay. So we know one of the parents is homozygous recessive. Okay. So she's going to be little h, little h. Okay. Now again, we don't know if the parent is going to be homozygous dominant or heterozygous. So for what we're going to kind of do 2 scenarios here. Okay, the first scenario is we're going to assume that the parent is homozygous dominant, and then we're going to look at the offspring. Okay. So we'll say that that parent is going to be big H, big H. Okay. So we do our Punnett square. Here we have big H, little h, big H, little h, big H, little h, and big H, little h. Okay. So that's one scenario. Okay. If we do the other scenario, oh geez, let's try this again. Okay, then if we assume that parent or the male is heterozygous, okay, we got big H over or little h, little h. Okay, and let's assume this parent is big H, little h. Okay, again, do our Punnett squares. We have big H, little h, little h, little h, big H, little h, and then little h, little h. Okay, so which of these 2 Punnett squares gives us offspring that all have straight hair? Okay. Would it be the one with the parent that's homozygous dominant or the parent that is heterozygous? Based off of what we see in our Punnett square. So in this particular case, it would be the parent that is homozygous dominant, right? Yeah. So the parent that's homozygous dominant is going to be this one here. Okay. So we see that all the offspring have straight hair, which is going to be the dominant one. Here, they all contain the dominant allele, so they would all be straight-haired offspring. So for that reason, we know that the parent had to be homozygous dominant. Okay. Here we see that half of the offspring are going to have straight hair and half are going to have curly hair. Okay. But that does not go hand in hand with what we see with our results. Okay. So we know this one cannot be the case. So the answer here is that we're looking at a parent that is homozygous dominant. Okay. Does this make sense? How to do a test cross? Anyone have any questions on how to do a test cross here? Okay. All right, so we'll go ahead and keep going here. Okay, so next one. So question 6 says, Sam grows red roses and yellow roses. So after letting them cross, he found that there were only red roses in the next generation, the F1 generation. Then he let the F1 roses self-fertilize. In the F2 generation, he noticed that some of the roses were yellow. Okay, which is most likely true about the yellow roses? Okay. So A, we have the yellow petals were a recessive trait. B, yellow petals were a dominant trait. C, yellow petals were incompletely dominant to the red petals. Or D, yellow petals were co-dominant. Right, so everyone's saying A, so, and that is correct. So the yellow petals are a recessive trait, and the reason we know is because it skips generations. Okay, we found it in that first initial parent generation. Okay? It was not present in the F1 generation, in the second one, okay? But it did show up in the F2 generation again. So it skipped generations, so we know this has to be a recessive trait. Cool. All right, question 7. So here we have a Punnett square again. So we have a male pattern baldness. Male pattern baldness is an X-linked recessive trait or recessive disorder here, labeled X little b. Okay. The dominant normal hair gene is X big B. So Jack has male pattern baldness, okay, and Jill is a carrier, which means she's heterozygous male pattern baldness. Okay. They have a child. So if Jack and Jill have a daughter, what is the probability that she inherits male pattern baldness? Okay. So let's go ahead and kind of work out the Punnett square here. Okay. And so if we're starting with Jack, Jack we know has male pattern baldness. So he's the genotype X little b Y. Because again, Jack is a male. Okay, so he's going to have a Y chromosome and an X chromosome. Okay. Okay, I am back. So hopefully this doesn't happen again. Hopefully I don't get lose my connection again. So let's try this one last time here. Okay. So, all right, now everyone able to see the PowerPoint again? Is everyone able to hear me now? Okay, cool. All right. So let's attempt this one more time here. Okay, so we talked about about Jack here being X little b Y. Okay. And then if Jill is heterozygous, okay, if she's a carrier, what would her genotype have to be? Right, so she'd have to be X big B, X little b. Perfect. So X big B, that's supposed to be a big B, and then X little b. Okay. So again, if we do the Punnett square here, we're gonna have X big B, X little b. Okay. This one we're gonna have X big B, Y. Okay. Then here we have X little b, X little b. And then here we have X little b, Y. Okay. Okay. So here we have they have a child. Okay. So if Jack and Jill have a daughter, okay, what is the probability that she inherits male pattern baldness? What do we think? So yes, Kimberly. So it would be about 50%. And so the reason we know that is because we're first going to look at just the possibilities of our female offspring, which are going to have 2 X chromosomes. So that's going to be these offspring over here. Okay, so these are going to be our daughters. And 50% of our daughters, one of our 2 boxes here, is going to have the male pattern baldness genotype here. So we'd have 50% here. Perfect. Okay, cool. All right, that's question 7. Question 8. So if a black mouse is mated with a white mouse and all the offspring are gray, what type of inheritance pattern is being displayed? Just incomplete dominance, simple dominance or recessiveness, codominance, or sex-linked? Right, this would be incomplete dominance. Perfect. So incomplete dominance is where you have a blend of the 2 phenotypes. Okay? So if you have black and a white, okay, they end up mating together and they end up forming offspring that are gray. Okay, last question here. So this one, the most basic of all of our questions so far. Okay, so here we're going to start with a Punnett square. So in mussels, okay, brown shells are dominant and blue shells are recessive. Okay? So a homozygous brown mussel mates with a blue mussel. So what is the probability that the offspring will be brown? Okay, so again, we'll go ahead and do our Punnett square. Okay, so we have a homozygous brown mussel. Okay, its genotype would have to be big B, right? Because that's the dominant allele. Okay, and then it also mates with the blue mussel, which is recessive. So if it's recessive, it has to be little b, little b. Okay, little b, little b. Okay. And so if we do our Punnett square, we should have big B, little b, big B, little b, big B, little b, and big B, little b. Okay, okay, so based off of our Punnett square, what is the probability that the offspring will be brown? Okay. And so we get 100%, right? Because all 4 of our possible offspring are going to have the dominant allele. So there's a 100% chance of having brown offspring. Okay, any questions from this, I guess from this chapter here? Okay. All right. So if not, one thing I do want to remind you of is the extra credit assignment. So remember, the extra credit assignment is going to be due today, okay, by 11:59 tonight. So that is located. That's located under the Chapter 8 module on the homepage, and you'll see 2 extra credit assignments, and you can submit those whenever you're completed with them. And so you can either email it to me or you can submit it directly onto Canvas. Either one is fine. So make sure you do that because those are each worth 10 total or 10 points each. Okay, so if there's no questions, we'll go ahead and jump into our lecture here and try to finish up this chapter. So Paul asked, how should we do the extra credit assignments if we can't print them? So what you can do is you can just write in the answers on Microsoft Word, because they're all Word documents. You can just put in the answers directly onto Microsoft Word, save it, and then just submit it onto Canvas. So that's probably the easiest way to go about it. The extra credit is, so Tim's question is the extra credit mandatory? It's not mandatory. So you don't need to do it, but it's one thing that will definitely help you if you're not doing so hot in the class already as it is. So definitely highly recommend it, because it'll also give you extra practice for any Punnett squares, the questions that you will see on the exam too. Yeah. So Kimberly says they're easy and would definitely recommend. So yeah, I would definitely recommend it too, if for nothing else, for the practice. Okay. Any other questions here? Okay. So Christian says, so I couldn't get the format when placing the allele on it, so I was wondering if I could print it on it. Yeah, so it's totally fine. So if you have any questions, feel free to email me and I can help you with that as much as I can. Okay, so we'll go ahead and finish up talking about X-linked disorders and kind of looking at some of those extensions of genetics here. And so we're just finish off by looking at this particular situation where we're looking at a red-eyed male, and we're again looking at flies with the red eyes being dominant and the white eyes being recessive. Okay. So here we have a red-eyed male crossed with a red-eyed red-eyed heterozygous female. So let's go ahead and try doing this Punnett square. Okay. So if we do the Punnett square here, oops. So if we have a red-eyed male, what would his genotype have to be? Okay, again, assuming red is dominant and white is recessive, what would his genotype have to be? X big R Y, perfect. So X big R Y, cool. And then for the female, if she's a red-eyed heterozygous female, what would her genotype have to be? Right, X big R, X little r. X big R, X little r. Okay. And then again, if we were to do this Punnett square, what should we come up with in this top left box? Right, X big R, X big R. Cool. Okay. What about the top right? X big R Y. Perfect. And then bottom left. X big R, X little r. Cool. And the last box, bottom right, should be what? X little r y. Perfect. Good. Okay. So now if we were to kind of analyze our Punnett square here. Okay. So what is the probability of offspring offspring with red eyes? If we're looking at offspring as a whole with red eyes, what's the probability? So the answer is 75%, right? So 75% chance of having offspring with red eyes. Again, so we see 1, 2, 3 of our 4 boxes having the red eye allele. Cool. And then the probability of offspring with white eyes would be what? 25%. Good. And again, that's just because one of our 4 boxes has only the recessive allele. Cool. All right. So now, what about the probability? Let's put probability of a son with red eyes. What's the probability of having a son with red eyes here? Right, 50%, because again, you're going to be looking at just the male offspring, okay, where we have our Y chromosomes. One of our 2 boxes has the red eye allele, okay? And then there was the probability of having a son with white eyes. Right, that'd be another 50%, because again, the other one of our 2 boxes has the red eye allele, okay? And then what about the probability of having a daughter with red eyes? Right, 100%, because again, 2 out of our 2 boxes here have the red eye allele. Perfect. Okay, so Angelina's question is, how do we get to 25%? So when you're looking at the offspring, right? So if we're looking at probability of having offspring with white eyes, okay, they are the recessive allele. Okay, that's the recessive trait. Okay, so if we look at box number 1, whoops, that's not what I wanted to do. Put this back in blue. Okay, so if we're looking at box number 1 here, okay, box number 1 is going to have 2 copies of the dominant allele. Okay? So they're going to have all red eyes here. Okay. Box number 2 has just the dominant allele here. So again, that one's also going to have red eyes. Okay. This third box is going to have 1 copy of the dominant allele. So that one will always also have red eyes. Okay. But this one here only has the recessive allele. Okay. So that one's going to have white eyes. Okay. So what we see here is that one of our 4 offspring, okay, one of our 4 boxes, will end up having white eyes. Okay. So we have a 1 in 4 chance of having white eyes in your total offspring here. Okay. Okay. So Stephanie's question was, why is it that we have a 100% chance for daughters? Okay. So in daughters here, so again, we're just looking at our daughter boxes. Okay, that's going to be these 2 boxes here, because we have 2 X chromosomes. Okay. So we see that this box has 2 copies of the red eye allele. Okay. This one has 1 copy of the red eye allele. Now remember, the red eye is going to be dominant. Okay. So for that reason, okay, both of these are going to have red eyes. So 2 out of our 2 boxes are going to have the red eye allele and eventually have the red eye trait. Okay. All right. Any other questions here? Okay. All right. So if not, oops. So this is what we would see with our Punnett square. And this is essentially what we already came up with here. So we found that we're going to have both red-eyed and white-eyed offspring. Okay? All the females that we have are going to have red eyes, okay? While 50% of our males are going to have red eyes, and the other 50% will have white eyes. Okay? Now, one kind of interesting thing that I wanted to bring you guys' attention to, actually, let me backtrack. Are there any questions on this question first? Any questions here? Okay. So if not, there is one thing that I do want to bring our attention to. It's kind of like an interesting little tidbit here. But when we're looking at males and females producing offspring, one kind of interesting question is, how do we know whether we're going to have a male or how we're going to have a female, and how who's going to be responsible for determining whether our offspring is going to be male or female? Okay. Now again, if we're looking at females, okay, females are going to have 2 X chromosomes, while males are going to have an X chromosome and a Y chromosome. Okay. So when a male and female have offspring, okay, the female is always going to be passing down an X chromosome because that's all she has available, right? She can only pass down X chromosomes because she doesn't have any Y chromosomes, okay? So she's always going to be passing down an X chromosome, while the male can either pass down another X chromosome, and so you would end up with a female offspring, okay? Or the male can pass down a Y chromosome, okay, which would end up resulting in a male offspring or a son. Okay, so for that reason, when it comes to producing offspring, the men are the ones who actually, or the males are the ones who actually determine the sex of the child. Okay, females have no responsibility in determining the gender. Okay, when it comes to determining the sex, all that comes from the guys. Okay? So, sorry, ladies, you guys aren't so lucky. That's all us. Okay. All right, so that's that. That's just one little thing, one kind of interesting tidbit I wanted to get out there. But essentially, remember, males are always going to have an X and a Y chromosome. Females will always have two X chromosomes here. Okay. So we looked at all kinds of different. So Kimberly said, "Don't blame us when you don't get your boy." Then, yeah. So yeah, it's never the female's fault. It's always our fault at the end of the day. So when it comes to X-linked disorders, right? We've looked at a few different types of X-linked traits when we were looking at flies. Okay, but let's take a look at in humans. How does this kind of play out here? Okay. So one example of a sex-linked trait or a sex-linked disorder is color blindness. Okay. So a certain number of our population has individuals who are colorblind, where they can't see specific colors. Okay. And so this is actually an X-linked recessive condition. Okay. And so it is possible for women to not be colorblind, but they can still be carriers for it, which means they're heterozygous. Okay. They're carriers because they have one copy of the dominant color site allele, so they can see colors normally, okay, but they also have one copy of the color blindness allele. Okay. However, because that color blindness allele is recessive, it ends up getting masked by the normal color site allele. Okay. So their genotype becomes X big B, X little b. So carriers can be found in sex-linked disorders. It can also just be found in regular old simple dominance and simple recessiveness, whereas just, again, you're dealing with another heterozygote here. But this is one particular type of trait that can exhibit some color blindness, okay, and some carrier traits here. Okay. So let's kind of do one more example here where we're looking at a male with normal color vision or color sight with a woman who is a carrier for color blindness. Okay, so let's go ahead and do this last example here. Okay, so if we cross a male who has normal color sight, okay, so if he has normal color sight, what should his genotype be? Right, so it would be X big B Y. Okay. And then the female who is a carrier for color blindness, okay, what would her genotype be? X big B, X little b, right. So we get X big B, X little b. Okay. We end up doing our Punnett square. Let me do this. Okay. And so we end up with X big B, X big B. Okay. Here we have X big B, Y. Okay. What would we have in this bottom left box? X big B, X little b, perfect. And then in the bottom right, we should have what? X little by, perfect. Okay. So that's what our Punnett square would end up looking like here. Okay. So we'll go ahead and just kind of skip the analysis of this guy for the sake of time here. So that would be the Punnett square that we would come up with. And I'll just kind of get to the point with this here. And so when we're looking at this particular trait here, okay, again, we see 3 of our offspring, or 3 out of 4 offspring having the normal color site allele, okay? X big B, X big B, X big B over here. Okay? But we have 1 offspring that has the potential to be colorblind, okay? We said X little b, Y, okay? So what we see here is that the female offspring here in this case, are capable of inheriting the color blindness allele from their mom. Okay, here we have X little b, and so here, some of our offspring are going to inherit, or some of our female offspring are going to inherit the color blindness allele. Okay, but as long as their father is normal, okay, in this case, okay, they will have normal color vision because that dominant allele will mask the recessive one. Okay, nothing new there. Okay, however, the tricky thing comes when we're looking at males. So in males can be born with color blindness even if both of their parents have normal color sight. Okay, and that can be if mom is heterozygous. So if she's a carrier for color blindness, she's going to be passing on 1 copy of the her X chromosomes, and if that copy happens to be the diseased X chromosome, okay, they get the X little b allele. Now, on the flip side, they're not going to be inheriting an X chromosome from their dad. They're only going to be inheriting a Y chromosome. Okay? And so they kind of stuck here, having just having the color blindness trait, okay, even though neither of their parents themselves are colorblind. Okay? But again, that's going to be because mom happened to be a carrier for the color blindness allele, okay, or the genotype. Okay, so for this reason, typically X-linked disorders tend to be more common in men than women. Anyone have an idea why, based off of what we've just done here, based off our Punnett square, why would X-linked disorders be more common in men than in women? Anyone have an idea? Right, so Jerry said because, and Kimberly, basically because the father only has 1 X chromosome, or the sons only have 1 X chromosome. Okay? So because males only have 1 X chromosome, whichever X chromosome you inherit from your mom, that's what you get. Okay? And that's it, you only have 1 chance at it. Where females have 2 X chromosomes, okay? And so if they happen to get 1 copy of the disease allele, they also have, a chance to inherit a 2nd copy that might not be the disease version, right? So they have 2 chances to get the dominant allele, okay, whereas males only have 1 chance, and that's it. Okay? So in this case, females got a little easier than men do here. Okay? So again, the key here, males only have 1 X chromosome, so they got 1 chance. Females have 2 X chromosomes, so they get 2 chances at it, at getting the dominant allele. Okay, very good. Okay, so now kind of shifting gears from our Punnett squares here. We looked at a specific set of laws last week in class. And one of the laws that we saw was the law of independent assortment, which showed us that the chromosomes, the different homologous chromosomes, are going to be inherited separately. Okay? So the homologous chromosomes are going to line up along the metaphase plate during metaphase 1. Okay? And they're going to line up on either side of the metaphase plate independently of anything else, right? So they're just going to line up randomly. Once they line up randomly, they'll eventually get pulled to either side of the cell randomly, and then you have an equal chance of inheriting either one of those chromosomes. Okay? Now, if we kind of extrapolate that a little bit further, okay, what that also tells us is that the genes on different homologous chromosomes are going to be inherited separately, which again makes sense. If we have a gene on chromosome 1 and a gene on chromosome 4, they're not going to have any impact on which one ends up getting inherited, right? They're going to be inherited separately from each other. Now, we can also kind of take that a little bit further and say that there are different genes on the same chromosome, okay, that can also be inherited separately, okay, as a result of crossing over. Okay. So genes will typically be inherited independently of each other. Okay. However, there is a certain case here where we're going to have certain genes and certain allele combinations that are actually not inherited independently of each other and are actually typically inherited together. And so this is where things get a little bit more complicated. So when we're looking at a chromosome is going to have anywhere from hundreds to thousands of genes all lined up along that particular chromosome. okay? And some are going to be closer to each other than others. Okay? And so for that reason, there's going to be certain genes that may undergo crossing over together. Okay? They may actually be moved together with one another and not be moving independently. Okay? They're going to be moving with one another. Okay? So we call those genes linked. Okay? So linked genes are genes that are physically close to each other on the chromosome. Okay? And so because of their close proximity to one another, they have they're more likely, I should say, they're more likely to undergo crossing over together, okay, and be inherited as a pair. Okay? So I'll show you an example of what I mean here. Okay. So here we are looking at crossing over just like we normally would here in prophase 1. We're going to have our homologous chromosomes line up. They're going to pair up their tetrads, and they're going to align all their genes together. They're going to attach to each other at the chiasma, and then they're going to undergo crossing over and swap their genetic material. And then keep in mind that this is going to be happening in multiple places along the chromosomes. Okay? So there's going to be several alleles that are going to get shuffled during this process, not just one like it's shown in the picture. Okay? But it's going to be happening all over the place where we're going to have crossing over. Okay? Now, like we said, linked genes are more likely to undergo crossing over together because they're close in proximity to one another. Okay, so for example, here we're looking at two different genes. We're looking at a gene for plant height and then a gene for the flower color. Okay, so here we have a tall plant with red flowers, okay, and then here we have another chromosome that has a short allele and then a yellow flower allele. Okay, but let's just say these two genes, okay, are located very close to one another. Okay? So because they're located so close to each other, okay, they're undergoing crossing over together if crossing over happens in this region. Okay? So if crossing over happens, both of those two alleles are going to get crossed over with one another. Okay? So they're not going to be moving independently. Okay? They're going to be moving together. Okay? And so if those chromosomes end up moving together here, okay, then we know they're not moving independently, and that violates the law of independent assortment. Okay? Does this make sense here? Linked genes could just undergo crossing over together because they're close to each other on the chromosome. Okay. Any questions here so far? Any questions? Okay. Okay, cool. All right, so again, they're just located physically close to each other, so they undergo crossing over. Okay. So that would violate the law of independent assortment. Okay. So again, if we're looking at two different genes, we're going to be looking at a dihybrid cross, and so that would be where we have our Punnett square with 16 boxes. Normally, if the law of independent assortment is not violated, okay, if all of our genes are being inherited independently of each other, okay, then we should see a 9:3 to 3:1 ratio. Okay. However, because the law of independent assortment has been violated, because we have genes that are moving together and not independently, okay, that normal ratio does not hold true anymore. And so we don't get this crossing over taking place. Or we don't get the normal ratio occurring here. Okay. And again, that's just going to be because our genes have been crossed over together and not independently. Okay. Now, if the genes were further apart, okay, then they would be more likely to undergo crossing over separately, okay, and so they'd be more likely to undergo independent assortment, and they'd be more likely to independently assort with one another. Okay. And that's where we would see that normal 9:3 to 3:1 ratio. Okay. Again, so that's going to be due to chromosomes that are not linked. Okay. This would be chromosomes that are not linked and they're actually far apart or far enough apart from each other on the chromosome. We're going to see that normal ratio. But if they're too close to each other, okay, they undergo crossing over together. And so we do not get this 9:3 to 3:1 ratio that we normally would get. Okay. So I'll just leave it at that. Okay. Any questions on linked genes and how they work here? Okay. All right, so the last topic I want to bring up to you, which is kind of interesting, is called epistasis. Okay. So epistasis is, I'll get to epistasis in a little bit, but one of the things that Mendel was analyzing when he was first looking at genetics is that he kind of made the assumption, okay, that every phenotype was controlled by a single gene. Okay? And for some situations, that's the case, But in most characteristics, it's actually multiple genes all working together in unison to give you your particular trait. Okay? So for example, eye color is not regulated by a single gene. Okay? Eye color is actually determined by about 16 different genes all working together to give you your specific eye color. Okay? So here we have multiple different genes all working together to help you form an eye color. Okay? And each of those genes would have their own sets of alleles that are going to be active here. Okay? So in most characteristics, most traits are going to be determined by multiple genes all working together. Okay? Another example here is we can have certain traits where genes can work synergistically with one another and actually help to form one particular trait. Okay, so for example, skin color itself would require at least 3 different genes, again, all working in unison with one another, okay, to give you your skin color. However, there are some genes that actually do work against each other. Okay, and so this is what's called epistasis. Okay, so epistasis is where one gene actually suppresses the action of another. Okay? This is different from dominance and recessiveness because dominance and recessiveness refers to one single gene and 2 different alleles. Okay? Here in epistasis, we're looking at 2 separate genes. Okay? One of them is going to be suppressing the action of one of the other genes. Okay? And so in order for a particular gene to get activated or to be expressed, it's usually going to depend on the gene that comes before it or the gene that comes after it during the activation process of that gene. Okay. So epistasis, we have one gene suppressing the action of another. Okay. So I'll give you an example here. Okay. So here when we're looking at mice, okay, there's 2 different kind of coat colors, right? There's the wild type coat color, which is the agouti pigmentation, which is about kind of like a brownish tan color. Okay. And that is going to be determined by the dominant A allele. So that dominant agouti pigmentation, is going to be dominant over the solid black color, which is the homozygous recessive. Okay? So if you're dominant, you're going to have the agouti pigmentation. If you're recessive, you're going to have the black color, okay? Which is kind of what we see over here. Okay? So just like normal, heterozygous dominant, homozygous would give you the agouti, okay? Homozygous recessive would give you the black color. However, so that's the A gene. There is also a C gene. So we have a C gene here, and if a mouse tends to be homozygous recessive for that C gene, so they have a little c, okay, that homozygous recessive C gene completely negates the expression of the A gene. Okay? And so all the offspring end up becoming albino. Okay? So it doesn't matter if you're big A, okay? So example here we have big A, little c, okay? You would be agouti, but doesn't matter here because we have the 2 homozygous recessive C alleles here. And so because you're homozygous recessive for the C allele, all of these offspring end up becoming albino. Okay? If you're big A, little c, if you're heterozygous for the dominant or for the agouti color, or you're homozygous recessive for the C gene, doesn't matter, you end up becoming albino. Okay? Same thing if you're little a, little c, doesn't matter, you become albino. Okay? So that C gene, regardless of what the A gene is, will always negate the effects of the A gene. Okay? And it will itself take over. Okay. Yeah. So in this case, the C gene completely masks the A gene here, okay? Assuming it's homozygous recessive. Okay. Now, if we're looking at homozygous dominant or heterozygous for the C gene, okay, there's no effect. Okay. The A tends to take over. Okay, but if you have homozygous recessive for the C gene in this case, the homozygous recessive takes over, and we negate the effects of the A allele. Okay. So because the C gene has the ability to suppress the activity of the A gene, we call this the C gene epistatic, okay, meaning that it's standing over or it's expressing its dominance kind of over the A gene. And so the A gene would be considered hypostatic, meaning that it's under power of the C gene here. And so again, because we have epistasis here, we end up getting a different genotypic ratio. And so we end up with 9:3:4 of a phenotypic ratio here. Okay. Meaning that we have 9 offspring with the agouti pigmentation, 3 with the black pigmentation, and then 4 with the albino pigmentation. Okay. Any questions on epistasis here? Does this make sense? Let me start with that. Okay. So all epistasis is just we have one gene that can have the ability to negate the effect of another gene. Okay. And so that's what we saw with the C. All right, so if there's no questions, we will go ahead and move on to, yeah, so this is where things do get a little bit more complicated. Okay. So as far as genetics, we are now done with genetics, and we'll go ahead and dive into chapter 9 here. Okay, so everyone able to see the chapter 9 PowerPoint? Okay, perfect. Okay. All right. So chapter 9 is going to be looking at how we're actually going to be looking at the structure of DNA, and then we're going to be looking on Wednesday and possibly next week, depending on how far we're able to get into the how we're actually going to be able to activate our DNA. Okay. So we looked in the past about how DNA works. We looked at how we're able to replicate DNA and produce identical cells. We were able to look at how we're able to produce offspring that are going to be genetically unique, okay, through meiosis. And then we looked at how genes can be inherited, okay, but we haven't really looked at the structure of DNA itself. Okay, so that's going to be what we're going to be starting with today. And so we're going to look at how DNA is built. And so we're going to start by taking a look at a few different scientists here. So in the 1950s, so about 100 years after Mendel did his work on genetics, there are a bunch of scientists that started to wonder how DNA is actually structured, how it's built, what does it look like, okay? And so the two kind of leaders of the pioneers trying to understand how DNA is built were these two scientists named Francis Crick and James Watson. Okay? And they also worked with a bunch of other scientists to help kind of identify the structure of DNA. Okay, so some of those scientists were Linus Pauling, Maurice Wilkins, and Rosalind Franklin. And what they did was they used a technique called X-ray crystallography. Okay? So they knew DNA was there, but they didn't know its full structure. They didn't know how it was built. Okay? So they used this technique called X-ray crystallography, which is kind of interesting because what you do is you take that particular molecule that you're looking at, in this case, DNA, you crystallize it, and then you shoot a bunch of X-rays at it. Okay? So if you got this crystal structure and you shoot a bunch of X-rays at it, the X-ray kind of particles are going to kind of bounce off based off of the structure of, the DNA molecule. Okay, and it kind of bounce off onto a sheet, and then it almost creates kind of like a shadow or a silhouette of structure of DNA. And so that was the first ability or the first technique that was used to help them understand how DNA was built here. Okay, so after these three scientists used their X-ray crystallography technique to help identify the structure, Watson and Crick then kind of used that data and then used it as kind of the last piece of the puzzle to help figure out the full structure of DNA. Okay. So then there was another scientist named Erwin Chergaff, okay, and what he determined was that there had to be 4 different monomers, okay, 4 different kind of sub things within DNA, okay, that helps to kind of give it its structure. Okay. And so one of the things that he saw was that 2 of the monomers, okay, were always found in equal amounts, and the other 2 were also found in equal amounts to each other. Okay. So that was Chargaff's rule. So 2 monomers were always found in equal amounts, while the other 2 were also found in equal amounts to one another. And what that told them was that those 2 sets of monomers had to have showed up in pairs. Okay, they must be coming in pairs with one another. Okay. And so we're going to take a look at what exactly he meant by that in a second. Okay. So before we get to that, so just keep that in mind. We'll come back to Chargaff's rule in a bit. Okay. So when we're looking at the nucleic acids, remember we're dealing with 2 different types of nucleic acids. Okay, one of them is deoxyribonucleic acid, or DNA, okay, which is over here. Okay. So deoxyribonucleic acid, and then the other one is ribonucleic acid, or RNA. Okay, so those are the 2 types of nucleic acids we're going to be dealing with. They look very, very similar. Okay. There's only one slight change here. Okay. In DNA or in deoxyribose, okay, we have just an H at the 2' carbon. So this is the 1' carbon, here's a 2' carbon. Okay. There's just a hydrogen attached to this carbon molecule over here, or this carbon atom. In ribonucleic acid, we're going to have a ribose sugar. Okay, and so here in the at the 2' carbon, we have an OH attached to the carbon here. Okay, so there's a little bit of a difference here, very slight, very kind of minimal difference, but a difference that makes a big, kind of a big change here. Okay, so those are the 2 nucleic acids we're going to be looking at in this chapter. Okay, DNA and then RNA. Okay, so when we're looking at DNA, we're going to look at the structure of DNA first. Okay? And so DNA is going to have 3 different parts to it. Okay? The first part is going to be a sugar, which is in this case the deoxyribose, this part here. Okay? So it's going to be a 5 carbon sugar. We have 1, 2, 3, 4, and then a 5th carbon up here. Okay? And it's going to have a hydrogen attached to the 2' carbon. Okay? Attached to the sugar, attached to the deoxyribose sugar, we're also going to have a phosphate group on one side, okay? And then we're going to have a nitrogenous base on the other side, okay? Which will be either adenine, guanine, cytosine, or thymine, okay? And so that's how DNA is going to be built, okay? With those 3 different parts to it. Okay? And so these are what these nitrogenous bases will look like. Okay, so here's our adenine, this is guanine, we have cytosine and thymine. Okay, so we have a purine. Okay, these 2 structures with 2 rings on it, or 2 attached rings, are adenine and guanine. Those are called purines. And then the cytosine and thymine, those are considered the pyrimidines, okay, because they're just a single ring. Okay, again, don't worry too much about that, but just that's just what their structure looks like. Okay, okay. So going back to Chargaff's rule, what was Chargaff's rule? We have A, each monomer of DNA is present in random amounts. B, 2 monomers of DNA were present in equal amounts, and the other 2 monomers were also present in equal amounts. Or C, the monomers were independent of one another. What do we think here? Yes, so Yeti's got this one right, and Mario, perfect. Right. So Chargaff's rule was that 2 of the monomers were going to be found in equal amounts, and the other 2 were also going to be found in equal amounts. Good. Okay, so when we're looking at DNA, okay, DNA is going to be kind of arranged in a double helix, and there's going to be these 4 monomers that are going to be making everything up. Okay, so then those particular monomers that Chargaff was talking about happen to be the nitrogenous bases, okay, the adenine, thymine, cytosine, guanine. Okay, so those are the monomers that hold DNA together. Okay, so, and they're always going to be showing up. Okay, so what we saw was that we were going to have an adenine, or A, that's going to be attached to the thymines, which are our T's. Okay, those will always be found in pairs. Okay, and then guanines on one strand of DNA are also going to be bound to cytosines on the other strand of DNA. So they're also going to be found in pairs. Okay, so for that reason, the amount of A's and the amount of T's, okay, are always going to be the same, and the amount of C's and the amount of G's are also going to be the same. Okay, so those 2 sets of monomers are always going to be found in equal amounts to one another. Okay. So adenines will always bind with thymines, guanines will always bind with cytosines. Okay. So A is always with T's, C is always with G's. Okay. And so this is what we refer to as complementary base pairs. Okay. So they're considered bases because these are all nitrogenous bases here, and they're complementary because they're always going to be found together. They're always going to be found paired up with one another. Okay. Again, A is always with T's, always with G's. Okay. So Kimberly's question, are they actually shaped like that, round and angled? No. So they're not actually shaped like this. This is just kind of a picture to just help you kind of visualize how they're going to be fitting together, essentially. Okay. But they're going to be bonded to one another, and I'll actually show you a picture of what that looks like in a bit here. Okay. So again, A is always with T's, C is always with G's here. Okay. So we have a question here. So we have a DNA sequence on one strand that is as follows. Okay. We have ATG, CAG, TA. Okay. So what will be the complementary sequence on the opposite strand of DNA? Okay. So go ahead and take a few seconds and see if you can figure this one out. So I'm seeing C's, and C is the correct answer. Yes. So we're going to have C's because, again, our A's are going to be binding with T's. Okay, so we have A here, so we're going to see a T on this opposite strand. Okay, here we have a T here, so we're going to find an A on the opposite strand. Here we have a G, so we're going to see a C on the opposite strand. C, so we're going to have a G on the opposite strand. A, we have a T, G with a C, T with an A, and then A with a T. Okay. So these strands of DNA are going to be complementary to each other, which means they're going to be opposites. They're going to have the opposite base pair on that particular strand of DNA. Cool. Everyone following me so far? Okay. All right. Yes, Kimberly, very exciting stuff here. Okay, so now we're going to take a look at the kind of the backbone of DNA. Okay, so we looked at the kind of, so if we take a look at DNA, DNA is going to be kind of shaped like a double helix, right? But if you kind of unwind it and make it into like a ladder formation, we just looked at the nucleotides, the nucleic acid bases, or the nitrogenous bases, which make up the rungs of our ladder almost. Okay, it almost looks like a ladder. Okay. So the rungs of our ladder are going to be made up by the nitrogenous bases, but now we're going to look at the legs of our ladder. Okay, we're going to look at the backbone of the DNA. Okay. So the backbone of the DNA is going to be made up by the phosphate and the sugar. Okay. In this case, the phosphate and the deoxyribose. Okay. So here what we're going to have is our phosphate and our sugar here, our deoxyribose, are going to be bonded to each other in a very specific manner. Okay. What's going to happen here is we're going to have our phosphate that's bound to the 5' carbon of the deoxyribose. Okay, so again, we have our 1' carbon, 2' carbon, 3, 4, and our 5' carbon over here. Okay, and so our phosphate is going to be attached to the 5' carbon. Okay. Now the phosphate is going to also be attached to the 3' carbon of the next nucleotide, and so this is what it would look like here. Okay. So for example, here we have our 1, 2, 3, 4, 5' carbon over here. We're going to have our phosphate, and then it's also going to be attached to the 3' carbon, okay, on the next nucleotide. Okay, so again, on this next one, we have 1, 2, 3' carbon, and so that's going to be where that phosphate will bind. Okay, so the phosphate actually gets bound to 2 different nucleotides at the same time, and that is what holds the 2 legs of your DNA or your DNA backbone together. Okay, and so that's going to end up forming a polymer of our nucleotides. Okay, and so these are going to be made by covalent bonds. Okay. Now, in between those nitrogenous bases, okay, we're looking at the A's and the T's and the C's and the G's that are also going to be bound to one another. Okay. The way that they're actually bound to one another is by hydrogen bonds. Okay. So hydrogen bonds are going to be used to hold your DNA nucleotides together, our nitrogenous bases together. Okay. So when we're looking at the bond between the A's and the T's, okay, they're going to be held together by 2 different hydrogen bonds, okay, one over here and one over here. Okay. When we look at the interaction between the C's and the G's, okay, those are all going to be held together by 3 hydrogen bonds, okay, 1, 2, and 3. Okay. Okay. So together, kind of the phosphates held together by the phosphates that are holding our nucleotides together, as well as the hydrogen bonds in between the 2 different strands of DNA and the 2 different nitrogenous bases. Okay? That is what's going to hold our DNA together. Okay? The covalent bonds making up the DNA backbone, and then the hydrogen bonds on the nucleotides in between the 2 strands of DNA. Okay. So we were to kind of just backtrack here. This would be one strand of DNA. This would be the other strand of DNA. Okay. And then you have the nitrogenous bases, okay, that are being bound together by these hydrogen bonds. Cool. Okay. All right. So now when we take a look at that DNA, okay, the DNA is going to be arranged in a right-handed double helix. Okay. So what that means is if you were to take your DNA and you could just kind of unwound it into your ladder formation like we see over here, right? So this is our DNA, okay, in its ladder formation. If we want to get into its normal kind of double helical formation, you'd essentially take your right hand and you'd twist and twist and twist and twist until you end up forming a double helix. Okay, so each of the strands ends up forming a helix, and so we call it forming a double helix. Okay, so it's a right-handed double helix. And then the other thing here that, about the DNA is it's considered the two strands being anti-parallel. Okay, so here's what I mean about it being anti-parallel. This is what it gets a little bit confusing here. Okay, so when we're looking at the deoxyribose sugars, right, we're going to have a 1' carbon, 2', 3', 4', and the 5' carbon. Okay, so the 5' carbon facing upward on one strand of DNA, while the 3' carbon faces downward. Okay, they're always going to be in opposite directions, one facing up, the other one's facing down. Okay, so in this case, in this particular strand, we have the 5' carbon facing upward and the 3' carbon facing downward. Okay. On the other strand of DNA, it's going to be reversed. Okay, so the 3' carbon is going to be facing upward. So here's our 1, 2, and 3' carbon facing upward. Okay, while the 4, 5' carbon will be facing downward. Okay, so they're going to be oriented in opposite directions. Okay, one strand facing upward, the other strand with the facing downward. Okay. So they're going to be parallel, but they're going to be moving in opposite directions. Okay, so even though the two strands are parallel to one another, and one end might have the 5' carbon on the bottom end, while the other strand has the 5' carbon on the top end. Okay, and then vice versa for the 3' carbon. Okay, so they're just parallel, but they're oriented in opposite directions here. Okay. So DNA strands are anti-parallel. Any questions on the structure of DNA so far? What questions do we have? Okay. All right, looks like we're okay. Okay. All right, so kind of moving forward here, we're going to look at RNA now. Okay. So RNA is ribonucleic acid. Yeah, so here it is a lot to take in. There's a lot happening, kind of all going on here. So I'll take a little bit of time to study with this, after class. Okay, so RNA. Okay, so RNA is going to be ribonucleic acid, and it is going to contain the same 3 overall components as DNA. Okay, we're going to have a nitrogenous base. Okay, we're going to have the 5' the 5 carbon sugar. In this case, it would be a ribose. And then a phosphate all attached. Okay. The difference between RNA and DNA is that the sugar here is going to be ribose instead of deoxyribose. Okay. We're going to have an oxygen and a hydrogen bound to the 2' carbon. Okay. So it's going to be a polymer of nucleotides again here. And so now when we look at RNA, okay, RNA is going to have 3 of the 4 same nitrogenous bases. Okay. We're going to have an adenine. Here we're going to have our guanine. We're going to have our cytosine. And then we're going to have our uracil. Okay. So in DNA, we had an A, a G, a T, and a C, right? A, T, G, and C. Okay. But in RNA, we do not have any thymines. We don't have any T's. Okay. In RNA, that T gets replaced with a U or a uracil. Okay. So RNA has A, G, C, and a U, okay, while DNA has A, T, G, and C. Okay. So there's a slightly different set of nucleic acid base or nitrogenous bases here. Okay. Again, the only difference is we do not have any T's and we replace them with U's here in RNA. Okay. And in most cases, RNA is going to be single-stranded, okay, which means it's only going to have a single strand of RNA compared to DNA, which is always a double strand. Or double-stranded. Okay. So as a question here, so in viruses, RNA can be double-stranded, kind of like DNA. Okay. So based off of what we know, which nitrogenous base would adenine most likely bind to in double-stranded RNA? Okay. So if we're looking at RNA, okay, what do we think A's would be binding to? C's, G's, T's, U's, or A's here. Okay. cytosine, guanine, thymine, uracil, or adenine? What do we think? So Kimberly says D, and D is correct, right? So in RNA, okay, if there's any interactions with A's, A's are always going to be interacting with U's. Okay? C's and G's are still going to be interacting just like they normally would, and that's all they're ever going to interact with. In RNA, because we don't have any T's, the A has to interact with the U's. Okay? That can only interact with uracil. Okay? So A's always interact with U's in RNA. Okay. Now, when we look at RNA, there's 3 different types of RNA, and we'll see these on Wednesday, most likely. Okay. And so the 3 types of RNA are going to be messenger RNA, or mRNA. Okay. The other one is ribosomal RNA, or rRNA. Okay. And then the third type is transfer RNA, or tRNA. Okay. So these are the 3 different types of RNA that we'll be looking at later on this week. Now, when we're looking at DNA, okay, DNA has to be packaged within a cell in a very specific manner. Okay? And so the DNA that we're going to have in our cells need to be able to be replicated, right? They need to be able to replicate it for a number of different processes and a number of different functions. And then they need to be then read in order to help us produce some RNA, which will then help us be able to produce some proteins. Okay? So because DNA needs to be able to do a lot of these different things, it needs to be able to be packed very tightly and protected and packaged in a very specific way within the cell. Okay. The challenge with that is that a single DNA molecule is very, very long. Okay, you can take a DNA molecule and you can stretch it out to about 2 meters in length. Okay, so that's about 6 feet long of just one strand of DNA. Okay, or one DNA molecule. Okay, so obviously we know our DNA is not going to be 6 feet long, but it has to be packaged in a very specific manner. Okay, in order to fit inside of the cell, and not just within the cell, but to pack it inside the nucleus of a cell, okay, for eukaryotic organisms. Okay. So we're going to look at the way it's going to be able to be packaged. Okay. So when we're looking at prokaryotes, like bacteria, like E. coli, for example, okay, their DNA is going to be in a circular manner. Okay. Their genome is going to be circular here, okay, and it's going to contain about 4.6 million base pairs, okay, 4.6 million nucleotides, all bound with one another. So if you were to take this circular genome for prokaryotes, their DNA would stretch about, about 1.5 millimeters. Okay. Now, 1.5 millimeters may not seem like that large, okay, but that is about 1 million times bigger than this cell itself. Okay. And so again, you have to pack that very large genome into a very small cell. Okay. So in order to do that, what prokaryotes will do is they'll undergo a process called supercoiling. And so in supercoiling, what you do is you basically take the circular DNA and you coil it extremely tightly until you package it in a very small formation within the cell, okay, within the nucleoid. Okay. So you basically twist it beyond the double helix. Now in eukaryotes, this process is going to be a little bit more complicated. Okay. And the reason it's going to become more complicated is because eukaryotic DNA is much, much larger. Okay. And so in order to package that DNA, the first thing that we're going to do is we're going to take our DNA, which we find over here, and we're going to wrap that DNA around a protein called a histone protein. Okay. So this would be our histone protein right here. Okay. And so you're going to wrap the DNA around it. Okay. That whole structure of a DNA wrapped around a histone protein is called a nucleosome. So you're going to do that with various regions of the DNA, okay? And so you're going to have these big, nucleosomes that are going to be connected to other nucleosomes by very short segments of DNA. Okay? Again, this is all one DNA molecule, okay? All wrapped around all of these different histone proteins. Okay? So you're going to have multiple different nucleosomes within a single DNA molecule. Okay? And so they're all going to be connected to each other, okay, by regions of DNA that are not wrapped around histones, okay, but very short regions of DNA. Okay. Then you're going to take all those nucleosomes and you're going to stack them and compact them very, very tightly together. Okay. And then you're going to coil it even further than you initially would, okay, until that DNA molecule condenses, and then we see our chromosome arranged like this. Okay. And so when we're in metaphase, that's going to be where the DNA chromosomes are the most compacted. Remember when we were looking at DNA, we were looking at our chromosomes being condensed. Okay, this is what we're talking about. Okay, this is how we're able to condense it. Okay, and so it becomes the most condensed during metaphase. And so when we're looking at our eukaryotic DNA, chromosomes have the ability to either decondense or condense. Okay, in interphase, those chromosomes are going to be decondensed, and so those are going to be stages where the chromosomes can be stained using various types of processes here. Okay? So you can see it under a microscope. Okay? So if we have very, very dark stains, what that tells us is that the DNA has been packed very, very tightly. Okay, and so if it's packed very, very tightly, okay, that's going to tell us that the genes are inactive. Okay, so we call that heterochromatin. Okay? So genes are inactive because there's not a whole lot of enzymes that can be able to get into that particular region. Okay. Now, if we have very light stains, okay, we're going to find that our chromosomes are going to be more loosely packed. Okay, and so as a result, our DNA is going to be more able to be activated, more able to be used here. Okay. And so we call that euchromatin, meaning that those particular genes are able to be activated. Okay. Okay. So that would be euchromatin. Okay. Okay. All right, and that is that. I think we'll go ahead and stop it there. Yeah, we're a little after, so we'll stop it there. Couple things I would just want to bring to your attention before we finish off for today. So remember, be sure to complete the extra credit assignments. Next week we'll be working on, next week we'll have exam 3. As of now, it's scheduled for Tuesday, but depending on the situation and how much material we're able to get through next week, I may push that off to Thursday. I will let you know as soon as we finish with class on Thursday here. But next week we'll be having exam 3. Okay, as of now, aim for it being on Tuesday, but it may get pushed to Thursday instead, and we'll figure that out as we go. Okay. Any questions on anything we've covered so far? And we'll kind of pick this up. So Kimberly's question, is there a study guide? There is a study guide up on Canvas. So that encompasses chapter 6, chapter 7, chapter 8, and chapter 9. Okay. So that is up on Canvas. It'll be in the exam study guides. So it should be available to you already. Any other questions?